Every day on my way to school, I always pass this river behind the village. The wastewaters of the village collect in the river, causing it to have a high level of bacteria. But at one point of the river is this one plant growing on top of the water. The plant is Ecornia crassicus, or what is known as water hyacinth in English. A plant known to be useful to the environment due to its photoreplicative ability, but also notorious for its invasiveness. It grows so fast, and when it gets into numbers so large, it will cause the retardation of the flow of water. When it covers the surface of the water, it may cause harm to the environment due to its purification effect. And when this happens, people will remove these plants, leaving it useless like they did in most rivers Surabaya, Indonesia. So, knowing the characteristics of this plant, of being able to grow in an environment with high level of bacteria, but also to be present abundantly, a question then came into my head. Can this plant be used as a cheap yet effective antibacterial agent? My name is Josiah from Indonesia, and I proudly present my research, a novel antibacterial agent from Ecornia crassicus or water hyacinth. This research aims to find out whether Ecornia crassicus has antibacterial agent. Ecornia crassicus grows in areas ranging from warm temperate deserts to rainforest zones. It is recorded that Ecornia crassicus is capable of growing by 17.4% dry mass each day. There were two bacteria used in this research as the subject, Escherichia coli and Staphylococcus aureus. E. coli is a facultative anaerobic, gram-negative, rod-shaped bacteria known to cause diseases such as diarrhea and kidney failure in humans. Staphylococcus aureus is a facultative anaerobic, gram-positive, round-shaped bacteria known to cause a wide range of diseases in humans, such as bacteremia and toxic shock syndrome. There are two questions that this research is intending to answer. First, does Ecornia crassicus possess any bacterial properties? And second, if yes, is it bacteriostatic or bactericidal? There were therefore two experiments conducted in this research, to find out whether Ecornia crassicus has antibacterial properties and to find out whether it is bacteriostatic or bactericidal. The first experiment uses ampicillin as positive control, and this water as negative control. The plant parts used is the stem and leaf parts. The first step of the first experiment involves the extraction of the plant using ethanol. The plant parts are first cut into pieces and then immersed into ethanol to dissolve the chemical constituents contained into the ethanol. Next, the mixture is then filtered and the filtrate is heated to evaporate the ethanol it contains. Next, the extract is purified through centrifugation and pasteurization. The extract is then diluted into three different concentrations: 75% concentration, 50, and 25. Next, the pilot dishes are prepared. The bacteria is spread on the other. The cylinder cups are installed. On each other plate, 5 cylinder cups will be installed. The samples are then introduced to bacteria. The other plates are then incubated for 24 hours. Afterwards, the diameter of the inhibition zones produced is then measured. The measurements of the diameter of the inhibition zone is then recorded and made into graph. This graph shows the raw data of the measurements of inhibition zones. This graph shows the average measurements of the diameter of inhibition zone. As we can see, among the three extract concentrations, the highest inhibition effect is produced by the 75% concentration. From the first experiment, we found that the Ecornia crassicus possesses antibacterial properties. Ecornia crassicus is known to contain a chemical known as quercetin, which inhibits DNA kinases, an enzyme which is responsible for the supercoiling of DNA. The supercoiling of DNA is involved in the cell division of bacteria in the reproduction process of bacteria. Quercetin therefore inhibits the reproduction ability of bacteria. The second experiment. First, the fluid of the inhibition zone is taken and then transferred into a new culture media. The new culture media is then incubated for 24 hours. After 24 hours, observation is done to see whether bacteria grew on the new culture media. The observation proves that bacteria present on the new culture media, that including that the Cornelia-Christmas is bacteriostat. From the first experiment, we found out that the Cornelia-Christmas has antibacterial properties. And from the second, we found out that it works bacteriostatic. There are many applications of the Cornelia-Christmas as an antibacterial agent which I would like to investigate in the future. A Cornelia-Christmas can be used as an ingredient in hand sanitizers, antibacterial ointment, as well as multimodal drugs. A Cornelia-Christmas is present very abundantly, so abundantly that it is harmful to the environment and to the people. And when that happens, people will remove this plant, making it useless. But through this research, this plant, which is previously useless with no value, can turn into something that is useful and even life-saving. That is the end of my presentation. Thank you for your attention.
This is not a simple research for me. This is more advanced research for the students, I think. One question, we know that a cornea crest in Indonesian name Eceng Gondok, right? Okay, Eceng Gondok. So first, why you choose this?
Well,
Why?
as I said, every day on my way to school I saw this. On the river, on the polluted river, I saw this plant growing on top of it, on top of a river which is polluted and has a high level of bacteria. So I was curious, how can this plant grow in an environment so harsh? So I investigated and do this research.
Okay. Yeah, maybe the first, the main important, because this is like water hyacinth is as biomarker for the water pollution. I think this is very useful. The second question is, I don't know. You have been trying to study the literature for the compounds for the antibacterial in echinacea receptor from the water hyacinth. You have been trying. You have been studying the literature. What's the compounds?
Oh, yeah. According to research conducted by Marita and others, Cornelia-Christmas contains this following chemicals.
Yeah.
And among these chemicals are flavonoids and alkaloids, which has antibacterial properties. And after further research, I found out that the component which has been investigated for its mechanism of action is quercetin, a flavonoid. And so I included it in my research as the mechanism of action, as the predicted mechanism of action of the Cornelia-Christmas.
Yeah, maybe the main the important compounds here, in my opinion, there's two alkaloids and anthocyanins.
Oh, the antibacterial.
Yeah, for the antibacterial. Sterols and terpenoids, they don't have the activity for antibacterial. Flavonoids have the antibacterial, but not more power more than alkaloids and anthocyanins. But if this anthocyanin have the nitrogen rings, you have to put in the alkaloids compounds. So just very carefully what's the type of the structure of these anthocyanins. You have to check in your literature. Okay, maybe this we can put in the class of the alkaloids. Okay, because if this is compounds echinacea receptor after antibacterial, it's maybe they have the more alkaloid for the secondary metabolites. Just for third question, this technical question. You use this research, where you use when you do this research?
In university in Surabaya.
University in Surabaya, yeah, because this is more advanced research, I think, for the students. So, good results. Congrats.
Next question. Ada? Apa you?
I have no idea to make a question because
Not your area.
Ini juga karena waktu kita sedikit molor, kita harus ngejar pesawat. Mungkin untuk Josiah cukup dulu. Terima kasih banyak.